All right, everyone, welcome back to the show. Get ready because today we are diving deep into one of the most fundamental processes in biology, mitosis.
That's right. We are tackling the intricate dance of cell division that Dr. Rosario so perfectly laid out in our BIO110 lecture.
And you know what? We actually convinced Dr. Rosario himself to join us for this deep dive.
Oh, yeah. He's right here with us today to help us break it all down.
Dr. Rosario, welcome to the show.
Hey, thanks so much for having me on here. So glad to talk about mitosis. Let's go.
Mitosis is one of those things, it seems deceptively simple at first, but the more you learn about it, the more amazing it gets.
Absolutely. For me, the lecture left me with this like a whirlwind of phases and proteins and checkpoints. I'm definitely still trying to wrap my head around it all.
Yeah, I feel that. How does it all work so perfectly most of the time?
I know, right? The fact that DNA can be copied with such precision is mind-blowing.
Right, but like Dr. Rosario was saying, it's not a perfect process.
Yeah, the analogy he used in the lecture about photocopying, that was a good one.
Yeah, it's good that you brought that up. So remember, when we're doing mitosis, we're making identical copies of cells, just like how you would photocopy a piece of paper. But the thing that happens with photocopies is they're not exactly the same as what you started with. That kind of happens with mitosis as well.
If you keep making copies of copies, those tiny little imperfections start to add up, and the final copy might end up looking way different than the original.
And that's kind of what happens with DNA replication. Those little errors can sneak in.
Exactly. Sometimes mistakes happen.
Yeah, so just also keep in mind that when we're talking about DNA replication, we're really talking about the S phase of prophase, right? We're not actually talking about mitosis at this point. So let's circle back. Let's talk about mitosis and what that is, what it does.
Okay, so mitosis is all about cell division, right? But you mentioned in the lecture, Dr. Rosario, that there are three main challenges when it comes to that whole process.
Exactly. First, the cell has to replicate all of its DNA. Make sure each new cell gets a full set of instructions.
Right, the complete blueprint.
Exactly, a complete blueprint. Then, second, it has to separate all those copied chromosomes and do it accurately. And finally, it has to physically divide into two new cells.
Okay, so mitosis is specifically focused on that second challenge, separating the DNA.
Exactly.
And you really emphasized how crucial it is to get that separation right.
It's absolutely critical. Think about it like building a house, but you only have half the blueprints. It's not going to work, right?
Yeah, or it's going to have some serious problems.
Yeah. In the same way, if a cell doesn't get a complete set of chromosomes, it can lead to some major issues.
Okay, so the stakes are pretty high for getting this right.
Yeah, so before we can talk about mitosis, let's start off with G2 of prophase.
So why is it so important to understand what's going on in G2?
Right.
Why even talk about it if it's not technically part of mitosis?
Well, you can't understand the race if you don't know where the runners are starting.
Makes sense.
So G2 is like the starting line for mitosis.
Okay.
At this point, the cell has already duplicated its DNA.
Yeah, so just keep in mind that means that we have replicated chromosomes and sister chromatids at this point.
So it's doubled it, but it's still all kind of jumbled together in the nucleus.
Exactly. It's like this big bowl of spaghetti in there, all tangled up.
Okay, so that's one problem the cell needs to solve. But also, isn't the DNA still trapped inside that nuclear membrane?
For sure, for sure. So there's still the nucleus that's there, and it's still shielding the DNA, still protecting all the DNA.
So there are two big challenges. All that tangled DNA and the nuclear membrane acting as a barrier. Mitosis has to overcome both of these to separate everything correctly.
Okay, so the cell definitely has its work cut out for it. So what's the very first step in this whole grand scheme of organization and separation?
Well, the first phase of mitosis is called prophase. This is where things start to get organized and the cell starts setting up for the big division.
Right, and it all begins with the centrosomes, if I remember correctly.
Yeah, those little structures that start moving apart.
You got it. So the centrosomes are like the organizers. They start moving to opposite ends of the cell.
And this is really important because this really establishes the two different regions that are going to become the two different cells.
And as they move, they extend these long protein ropes called microtubules.
And those microtubules are going to be super important later on, right?
Yeah, these microtubules are the thing that allows migration and movement.
So we've got the centrosomes setting up the two sides. But what about the DNA itself? What happens to all that tangled spaghetti during prophase?
Well, during prophase, the DNA starts to condense. It coils itself up super tight and starts to form those X-shaped structures that we call chromosomes.
Oh, right, like the classic picture of
So
chromosomes.
it's like taking that jumbled mess of yarn and winding it into neat, organized ball.
Exactly. Condensing the DNA makes it much easier to move and separate accurately. And this whole process isn't just random. There are specific signals in the cell that tell the DNA when to condense.
It's amazing, like a perfectly choreographed dance. Everything has to happen at the right time and in the right place.
Totally. But I'm a little confused. So the chromosomes are condensing, the centrosomes are moving apart. What about that nuclear membrane? Doesn't that need to go away at some point?
That's a great question. And that's exactly where we're headed next. Prometaphase is where things get really exciting because that nuclear membrane, well, it's not going to be there for long.
All right, so where we left off, the cell is prepping for division in prophase. The chromosomes are all condensed, the centrosomes are moving apart. But what happens to that nuclear membrane?
Yeah, that's a big question. I mean, doesn't that protect the DNA? It just disappears.
Well, it does protect the DNA, but it has to get out of the way at some point. And that's where prometaphase comes in.
Yeah, so when we're talking about prometaphase, we have the disintegration of the nuclear envelope, we have the exposure of the chromosomes, but that is good for us because that means that we can now use things like microtubules and centrosomes to pull them apart.
Okay, so what happens in prometaphase?
Basically, that nuclear membrane breaks apart. Disintegrates, really. It breaks down into these tiny little vesicles.
So the barrier between the nucleus and the cytoplasm, it's just gone.
Yep, gone. And that allows the microtubules to reach the chromosomes.
It seems pretty risky, though, right? To just let all that DNA hang out exposed like that.
It definitely is a vulnerable phase. Things have to happen fast and accurately.
But now, the microtubules from the centrosomes can finally attach to the chromosomes, right? And this is where those kinetochores come in.
Right. Those tiny little Pac-Man proteins. I still can't get over how important those things are and how small.
They're amazing. Each sister chromatid, remember those are the identical copies of a chromosome, has a kinetochore. It's located right at the centromere.
The centromere, that's that central region where the two sister chromatids are joined together, right, to make that X shape.
Exactly. And the kinetochore, it's not just like a passive attachment point. It actually acts like a little motor, pulling the chromosome along the microtubule.
So it's like a train on a track. The kinetochore is the engine, and the microtubule is the track.
That's a great way to think about it.
Yeah, and I would actually just add a little bit to that because it's such a great analogy. It's not just like it's a track, it's more like the kinetochores are chomping away at the track, pulling the chromatids apart.
But remember, there are microtubules coming from both poles of the cell.
So each chromosome is attached to microtubules on both sides.
Oh, so it's kind of like a tug of war then.
Well, not exactly a tug of war. The microtubules aren't pulling in opposite directions. They're actually pushing against each other.
Pushing. Okay, that's a little confusing. So if they're pushing against each other, how do the chromosomes separate?
It's this pushing that creates tension. And it's that tension that forces all the chromosomes to line up right in the middle of the cell.
Oh, right. And that's what leads to that classic image we always see. The chromosomes all lined up neatly in a row. Metaphase.
Metaphase, that's the easiest phase to recognize, right? When you see that perfect line of chromosomes.
Yeah, exactly. It's actually the longest phase of mitosis, too.
Really? That's interesting. I guess it makes sense, though, if the cell needs to make sure everything is lined up perfectly before it starts separating things.
Exactly. And that's why there's this important checkpoint during metaphase.
Oh, right, the M checkpoint.
The M checkpoint is like the cell's quality control system. Before going any further, the cell double-checks everything. Make sure every single chromosome is attached to microtubules from both sides and that they're all lined up correctly.
So it's like a final dress rehearsal before the big performance.
Yeah. Make sure everything is in its place and ready to go.
Exactly. If something's not right, the cell can actually pause mitosis at this point and fix the problem.
How does the cell finally separate those sister chromatids?
Yeah, all that tension has got to go somewhere, right?
Well, that's where we move into anaphase, and this is where the real action happens. Those cohesin proteins, remember, they're like those little clamps that hold the sister chromatids together.
Right, the molecular glue.
Well, in anaphase, those clamps get released, and now the sister chromatids are free to separate. So now we have two separate chromatids for each chromosome. What happens to them?
Well, those microtubules that were pushing against each other, remember? Now they help to pull the chromatids apart. And the kinetochores are still doing their thing, pulling those chromatids along the microtubules toward opposite poles of the cell.
So the cell is actually getting stretched out at this point as those chromatids move apart.
That's right. And with the chromosomes heading to their final destinations, we're getting ready for the final phase of mitosis.
Yep, here we are at telophase and cytokinesis.
Well, it's time to start rebuilding, and that's what telophase is all about. Remember that nuclear membrane that dramatically disappeared back in prometaphase?
So like the walls of Fort Knox just crumbling.
Yeah, when it was just dissolved.
Well, now it starts to reform around each set of chromosomes.
So the DNA is protected again.
Exactly. And those chromosomes, they start to uncoil and decondense.
Makes sense they need to be accessible again so the cell can actually use them.
Right, like unpacking all those boxes after a big move so you can finally find your stuff.
Exactly. And while all of this is happening, the cell itself is getting ready to divide. This is the final step. Cytokinesis.
Cytokinesis, that's where the cytoplasm actually divides,
Yeah.
right? And that's how we end up with two separate daughter cells.
Right, two complete cells.
So the process will end with 2 cells, but let's just keep in mind that animal cells and plant cells have to do this very differently.
It's incredible how much has to happen to create 2 new cells.
Yeah, it really makes you appreciate all that complexity at the microscopic level.
I know, it's amazing. Well, Dr. Rosario, thank you so much for walking us through all the steps of mitosis.
Yeah, of course. I'm happy to talk about BIO110 topics anytime.
This was seriously helpful. I feel like I actually understand mitosis now.
Me too.
Awesome. I mean, that's the goal, right?
And now I can't stop thinking about all those cells dividing in my body right now.
Right. Like every time we get a cut or a plant grows taller, it's because of this amazing process of cell division.
Exactly. Mitosis is literally happening all around us all the time.
Well, that's it for our deep dive into mitosis. Thanks for listening.
And a huge thanks to Dr. Rosario for joining us.